women's health needs innovation and creativity. It needs everybody to look at it in a new way.
There is a difference in the biology.
There is just this massive data gap when it comes to the female body.
It's not a women's health problem, it's a problem.
The immune responses are very, very different. In response to a pathogen threat, a male immune system will launch the Army. The female immune system will launch the Army, the Navy, the Air Force, and the Marines.
This past June, WHAM hosted a celebration in D.C. for the 30th anniversary of the Revitalization Act of 1993.
30 years since women and people of color were finally included in NIH clinical trials.
I think it's time we do something. An anniversary should re-energize with a purpose.
3Not30. We don't want to have to wait 30 more years for many great things to happen.
Women's health research isn't just good science, it's a good investment.
We need to affect long-term change. We just can't invest blindly.
I'm going to be signing the presidential memorandum to establish the first ever White House initiative on women's health research.
Thank you, WHAM.
It's up to us, all women, to create the changes that we set forth today so that women and society flourish.
We can do this in 3 years. Let's not wait 30.
Good afternoon, and thank you so much for joining us. My name is Kiralee Lee, and I'm the founder and CEO of WHAM, Women's Health Access Matters, a nonprofit organization focused on the economic impact of accelerating funding for research and business investment in women's health. Welcome to the third dialogue in our 3Not30 Roundtable series. We are hosting these to kick off this year and share our objectives and strategy for achieving our 3Not30 goals. These dialogues will include expertise, knowledge, and passion, with a focus on changing the status quo of women's health. This past June, WHAM convened leaders from across the ecosystem in recognition of the 30th anniversary of the NIH Revitalization Act of 1993, commemorating the landmark legislation that initiated the inclusion of women and minorities in clinical trials and research, but also emphasizing the ongoing imperative for progress in women's health. These preeminent thought leaders committed to driving significant, impactful, and sustainable change in the next 3 years, not waiting another 30, creating the imperative of change in 3Not30. Developed by leaders during our June event, the 3Not30 initiative focuses on 3 ambitious goals: double the budget, improve diversity and access to clinical trials, and mine the data. These goals outline actionable steps to accelerate women's health research, innovation, and investment. First, double the NIH budget, the segment that reflects the 4 disease areas we studied in the WHAM report, focusing on diseases in which research is underfunded. This represents $350 million, which when doubled, brings a return of $14 billion to our economy. We also seek to double the amount of investment from VCs and other sources for funding for women from 2% to 4%, channeling more resources into business focused on the health of women and unlocking the substantial untapped growth opportunities in diagnostics, treatments, and prevention for women's health. Second, improve diversity and access to clinical trials. We need to increase diversity and equity in clinical trials by increasing the number of Black, Latina, and other underrepresented women participating in clinical trials. WHAM will support access to clinical trials and break down barriers, thereby enabling the enrollment of women and support our partners who are working to change this current dynamic. The third goal is mine the data and create a WHAM accountability index to identify through all means those who meet and those who do not meet the criteria set by NIH to report analyses by sex. Data is key to WHAM. And not only will we continue to drive to have more data on women and be sure that we are reflecting the basic differences of sex as a biological variable, but we're also turning our attention today to autoimmune disease and ambitious, actionable goals for the next 3 years. With autoimmune disease disproportionately affecting women, comprising 80% of patients, it is crucial to address the substantial gaps in awareness, funding, and innovation surrounding these chronic conditions. Esteemed leaders in this field will delve into the current landscape of these female-based and female-biased conditions and the critical research being done on sex-based differences in the immune system and share in an insightful dialogue on these 3Not30 goals, how will we achieve them, and how we can support each other to create sustainable and dramatic change to improve the health and well-being of women, their families, and society. Joining me today are exceptional leaders, Dr. Montserrat Angera, Associate Professor at the University of Pennsylvania, Dr. Lori Frank, President of WHAM, and Dr. Erica Ullman-Sapphire, President and Chief Executive Officer of La Jolla Institute for Immunology, Dr. Vicky Shanmugam, Director of the NIH Office of Autoimmune Disease Research in the Office of Research on Women's Health. With that said, I would now like to turn our attention to Dr. Erica Ullman-Sapphire, who will offer the perspective of not only an esteemed researcher herself, but also as the first female president and CEO of the La Jolla Institute for Immunology, which is one of the top 5 research institutes in the world focused on the study of the immune system. It was under Erica's leadership and through the partnership of WHAM that LJI launched the first of its kind Center for Sex-Based Differences in the Immune System, which focuses on how disease affects men and women differently. Welcome, Erica. And I wonder if you can speak to the evolution of research on autoimmune disease over the last 30 years, and specifically women and autoimmune disease. And what role does LJI play locally, nationally, and even globally?
Well, for thousands of years, people have understood that the body had an ability to heal, to fight against hidden threats from the outside world. And it had the ability to learn from those exposures how to be better the next time. It wasn't until the 1950s that we realized that the immune response could be directed against our own body as well as something foreign. Since then, we've learned that in general, autoimmune disease results from an interplay between a genetic predisposition and environmental factors. For the last 30 years, in the mid-1990s, it was common knowledge among clinicians that MS, multiple sclerosis, an autoimmune disease, was about twice as common in women than in men. Today, that ratio is closer to 3-to-1. And many saw that sex discrepancy in the disease as an inconvenience. And hardly anybody asked the question of why. And to compound that information gap, often to avoid the hassle of dividing their data by sex, researchers that were working with animal models might order only male mice or only female mice. And we know now that that sex discrepancy didn't end with MS. Women are generally more susceptible to autoimmune disease, sometimes by a factor of 10 or more, like Hashimoto's and Sjogren's. When at least some researchers started digging into the reasons why autoimmune disease predominantly affects females, their work began to open up fresh ways of thinking about conditions as diverse as heart disease, Alzheimer's disease, and Parkinson's disease. Now, these are not strictly speaking autoimmune disorders, but we now know they do share similarities. A lot of research is focused on sex hormones, which regulate the immune system through multiple pathways through receptors on immune cells. So it's not just your genetic encoding as XXXY, but the hormones that influence what's happening in your cells. Those hormones can explain why some people with autoimmune disease actually see their symptoms improve or worsen when hormone levels surge or drop, such as during puberty, pregnancy, and menopause. For example, in the second half of pregnancy, women with MS get about a 70% reduction in their symptoms and relapses. They feel great, but those symptoms return after giving birth. Women with lupus don't get the same reprieve. Now, more recently, others started suspecting that the sex chromosomes themselves were the key to the reason. Where every cell in our body is either XX or XY, and X is essential to life, and X encodes lots of different immune genes. But to avoid the double dose, much of that second X in a female cell is wrapped in a long RNA called exist that associates with 80 different proteins. It's a large complex. And it silences about 75% of the activity of that second X. Males don't have this complex because they only have one X. And researchers have recently discovered that many of these proteins trigger the production of autoantibodies. The newest theories now point to a complex interplay between hormones, sex chromosomes, and other physiological factors. And if we dig into the molecular basis by which autoimmune disease triggers and how it's different between different people, we can find the treatments that we need for all kinds of diseases. In 2016, NIH created a policy to mandate the inclusion of sex as a biological variable in research design. Now the topic of sex differences in autoimmune disease and other kinds of disease is finally making its way to the main stage with new funding opportunities and more discoveries that are throwing open the doors to what we need to know to bring better health to both men and women.
You know, it's just so enlightening when you explain it that way. And of course, we're going to hear more about access from Montserrat in a little bit. You know, when you joined LJI, I don't think there was the same emphasis, obviously, as there is now, or the same interest in studying sex as a biological variable. How has LJI changed over the years, since you've been there and identifying and addressing the unique needs of women and autoimmune disease?
Yeah. That's a great story. So LGI focuses on the immune system, and we have 25 expert faculty in different areas of the immune system. And when I became president, we went through a year-long bottom-up strategy process of asking the faculty, you know, what is the most impactful thing that you want to do in research? What do you want your legacy to be? What are you uncovering that intersects with the other faculty? How to be, instead of 24 laboratories in a building, how do we align the vectors to make the greater leaps that we need for human health? And what emerged from that, from all of our researchers, is they were beginning to discover, in whatever disease they happen to be working on, differences between male cells and female cells, and male mice and female mice, and male people and female people. There were landmark papers that were starting to come out from, we have these national databases where we make this immune information available to all researchers, like the database of immune cell epigenetics, what things are turned on and off and what levels. That houses a massive data set that results from our La Jolla Institute investigators' immunoprofiling efforts to see how variations in DNA sequence and sex-based differences impact gene activity. And what our researchers found is that there were 1,700 genes that are launched differently, whether the cell is male or female, that come from even chromosomes that are not X and Y. So they're two very different biological populations that are launching biological activities in very different ways. And if you don't understand that, you might spend all of your time studying a cell line that is only male, in which there is no exist at all. You would never see it. Or a cell line that are only female, where you don't understand the differences. Through this strategy process, we realized that this was something underlying everybody's research in the building, and was something that influenced how we respond to infectious disease, cardiovascular disease, cognitive disease, and also autoimmune disease. And it became a guiding force to unify these efforts and build more research in this area. And that's our Center for Sex-Based Disease. That center that we built is now putting a laser-like focus on understanding the things that have been overlooked for way too long, the precise factors that underlie sex-specific variables in our immune systems, and mining those differences to improve women's health. That could be the role of certain immune cells or small metabolites that link inflammation to neurodegenerative diseases in men or women, or autoimmune diseases. And it could be genetic differences. It could be the differences in transcriptional levels of things, how much of one protein or one gene versus another. And as the mechanisms behind where these autoimmune diseases come from become clear, so do the opportunities for treatments and one day a true cure. You know, already therapy based on engineered cells has given some people with autoimmune disease, like systemic lupus erythematosus or idiopathic inflammatory myositis or systemic sclerosis, a new lease on life, free from fresh symptoms or treatments 2 years since the treatment.
It's so exciting, and I'm so constantly impressed with not only your knowledge but your leadership in really transforming, I believe, or imprinting at LJI the need to be studying both men and women, female and male cells, which, of course, is something that our next panelist, who's actively engaged in research as an associate professor at the University of Pennsylvania, Dr. Montserrat Angara, has also been very involved in. And we're just thrilled to have you with us. I wonder, Montserrat, if you can share a little bit about, obviously, excess, which we're all fascinated by. A lot of people might not know, what is it in the RNA that you're working in that might lead to greater autoimmune disease in women? And what does all of the research that you're currently delving into, how is that going to transform autoimmune disease?
Yeah, absolutely. Thanks, Carolee, for this opportunity first to be with this awesome group of women and to talk about this topic that I'm so excited about. So in my lab, so just a little bit about my training background. So I, as a postdoc, investigated X chromosome inactivation. And so this is something that is studied by a number of different labs across the world. Like, how do female cells in the early embryo decide which X chromosome to silence, right? So female mammals, we have two X's, either mom's copy or dad's copy. And in cells of the early embryo, in each cell, this decision is made either to shut off the dad's copy or the mom's. And then there's a memory of this silencing that takes place with every round of cell division. So as these embryonic stem cells are going on their journey to become a neuron, to become a cell of the immune system, they're remembering which X that they've silenced. So the majority of labs in the X inactivation field study this process in the early embryo. And when I started my lab at Penn, I had this training in stem cells and X chromosome inactivation, but I wanted to do something different. I wanted to specifically look at diseases that impact women where there could be problems with this inactive X chromosome. And so I started doing Google searches, right? A lot of literature searches, and I kept coming across autoimmune disease. And it really struck me that there are so many patients who are biologically female that are affected by this disease. And the literature, there was full of review articles that would point to the sex hormones as being sort of the end-all reason as to why there is this sex imbalance. And a lot of these reviews were written by people in the fields of immunology and autoimmune disease without knowing the epigenetic mechanisms that are involved in X chromosome inactivation. So I was coming at it from a completely different perspective and wondered, wait a second, there's got to be stuff happening with the inactive X chromosome. Could the silencing of that inactive X, and knowing what I know as a researcher trained in the field of X inactivation, could there be perturbations in these silencing mechanisms that then contribute to altered dosage of X-linked genes, which is observed in a lot of autoimmune disease patients, such as lupus and scleroderma. So that's really how we started getting into this field, to look at how this exist molecule. So I just want to take a moment and talk about my favorite RNA, which is this long non-coding RNA called exist. And it's an absolutely fascinating molecule. So I hope I can get everyone excited about RNA and particularly exist RNA. So this is a very long RNA. It's about anywhere from 17 to 20 kb in size. So it's a really, really long RNA, and it's never made into a protein. So it's never translated, and it functions as an RNA. So then you might be asking, well, what does this RNA do? This RNA has the ability to transcriptionally silence a chromosome. So in female cells, it turns off either the maternal or the paternal copy of the X chromosome. And what's so cool, in my opinion, about this exist RNA is that if you take it and you put it on an autosome, say chromosome 21, for example, it will shut off that chromosome too. So it has this absolutely fascinating ability to recruit epigenetic modifiers in a variety of different proteins to come in and transcriptionally silence an entire chromosome. So to me, that just just always blows my mind. So I hope to convey my excitement and enthusiasm for this RNA molecule.
Well, you definitely have. I mean, I wish I knew more about the science and could appreciate your excitement and your enthusiasm for the long chain of the RNA that you're explaining exists. And it's for those of you, it's XIST, which has been written about, and Montserrat has been written about and quoted widely recently, spoke recently out at LJI at a seminar, and will be speaking with Vicky in April at a conference that she's hosting. So what's its implications then? I love your enthusiasm, I love your excitement. And one of the things that occurred to me as you were talking is it sounds like there isn't enough collaboration between different sciences, different parts of the scientific community, because you kind of referenced that it was only your genetic background, if I got it right, that allowed you to perhaps see or examine this in a different way. Is there enough collaboration between researchers on this kind of issue?
Yeah, so I think the collaboration, the excitement, enthusiasm among scientists is absolutely there. It's more of like a language problem. So individuals that study immunology, it's very involved, and they're kind of speaking, let's say, I don't know what's another language. Let's say they're speaking Spanish. Mandarin, exactly. They're speaking Mandarin, and people who do epigenetics are speaking Italian, right? And so it's oftentimes really hard to speak both languages at the same time. I'm trying to become bilingual, right? So in my lab, we investigate epigenetic mechanisms in this context of autoimmune disease. And so my background in epigenetics, I've been doing a lot of reading, a lot of learning about the immune system and how it works. That's not something that I was originally trained in. It's just like when you pick up a second language or you pick up a third language. But absolutely, when I talk to my colleagues in immunology departments, they get so excited about the things that we're doing and the approaches that we're doing, and they want to collaborate. They're like, do you want cells? Do you want these mouse lines? Do you need access to patients? So the scientific community is absolutely happening. It's just a steep learning curve.
Fantastic. Well, we're excited for you and for all of us who will benefit from the great work that you're doing. So thank you for the exploration and for the passion. And with that said, I'd now like to turn to Lori Frank, who was a lead author on the WHAM report, and of course now is the president of WHAM, which I'm thrilled about. Lori, so I know we've talked a lot about the impact of the WHAM report on achieving our 3Not30 goals, particularly in terms of autoimmune disease, which can cause women to leave the workforce, as we've talked about, at very early stages and ages. What were some of the questions that you and your team were exploring? And talk to us a little bit about how the report brought attention to those disparities that exist in women's health, and how will it continue to impact the dialogue that is taking place?
Great, thanks for that. So first I'll set the context for the work. WHAM had contacted the RAND Corporation about examining downstream socioeconomic consequences of increased funding for women's health research. And the idea was to look across 4 therapeutic areas: autoimmune, but also oncology, cardiology, and neurology. I was at the RAND Corporation at the time and assembled a team, including health economists, so that we could then identify specific conditions within each of those 4 therapeutic areas to use as the basis for our modeling work to answer the question about what are the downstream consequences of increased funding. So we used the same criteria, looking across all those areas, and centered in on Alzheimer's disease and related dementia in brain health, lung cancer in oncology, coronary artery disease in cardiovascular, and then rheumatoid arthritis within autoimmune. Together, those conditions provide a really interesting complementary view of the impact that funding on women's health research specifically can have, especially because the diseases span a lot of different situations, including who is affected and when. So in the case of RA, we're talking about individuals who are affected often during prime earning years. So their workforce participation became part of our questions in that particular report. As you can imagine, in some of the other analyses, we were looking at impact on caregivers. So for example, for dementia. And those reports can be found at the whamreport.org, but also additional details are available on the RAND website. So just to give some of the numbers for RA, the percent of the extramural NIH portfolio that was woman-focused when we sat down to figure that out was a bit less than 7%, 6.6% for the years that we examined. And so we looked at what that translated into in terms of dollar amount. That was $6 million. And then we doubled that to see what that downstream consequence would be. And RAND used very conservative assumptions about the downstream health improvement that was the basis for the model. But even so, the return on the investment for our economy of increasing that 6 million investment by a factor of 2, so doubling it, could lead to 174,000% return on investment. So that's an awfully big number. And it's a huge return, practically speaking. So in terms of the impact that that work has had on the 3Not30 goals that you referenced, clearly we all saw the value and the actual value of a relatively small increment in funding in terms of economic return, and also in terms of the value to the quality of people's lives, which was something else that we examined.
You know, it amazes me when I know these numbers well, obviously the 7%, it was one of the smaller numbers, and yet women are 52% of the population, 51% of the workforce, own 60% of the wealth in the country, make 85% of the spending decisions, and make 80% of the healthcare decisions. Women are not a niche market. I am so thrilled, as we all are, that President Biden has decided that women's health research, as WHAM started early on, also wondering about, could change the economics of our country if more money and more research dollars and more investment in business was in fact allocated. So we'll get to that in a moment. But when I hear you, Lori, talk about the 7% and the return then to the economy by doubling a very small investment of $6 million, it reminds me of how far actually we've come. And it's only a couple of years actually since the WHAM report has been completed, and we still have one report left. So thank you for all the great work that you did in bringing us to this point. Vicki, I'd love to turn to you as the distinguished inaugural director of the Office of Autoimmune Disease Research in the Office of Research on Women's Health. And we know that there's so much that can be done to accelerate scientific discovery for these chronic conditions, which so disproportionately impact women, as we've been talking. Can you provide an overview of the Office of Autoimmune Research? Because it's a relatively new one, and what its objectives are, and also what inspired you to take on this new role, since you had a pretty stellar career, I think, at George Washington.
Oh, thank you, Carolee. And I'm so delighted to join you today at this roundtable and to meet all of your members. So as many of you know, back in 2022, the National Academies of Sciences, Engineering and Medicine conducted an analysis of the NIH research efforts pertaining to autoimmune diseases and published their reports in a report entitled Enhancing NIH Research on Autoimmune Disease. And based in part on some of the findings from that report, Congress then, via the Joint Explanatory Statement in fiscal year 23, through their Consolidated Appropriations Act, directed the establishment of the Office of Autoimmune Disease Research, known as OADR, within the Office of Research on Women's Health. And in establishing the office, they gave some direction as to what they envisioned with this, and that was that they envisioned coordination of a multi-institute and center strategic research plan. I will advise you the nomenclature has changed a little bit at NIH since those words were written, so it's now referred to as an NIH-wide strategic plan for autoimmune disease research. They asked that we identify emerging areas of innovation and research opportunity, that we coordinate and foster collaborative research across the institutes and centers, that we do a landscape analysis, so evaluating the NIH autoimmune disease research portfolio both at baseline and annually, and that we provide resources to support planning, collaboration, and innovation, and a kind of final goal to develop a publicly accessible centralized repository for autoimmune disease research. So a real very lofty series of tasks that we've been asked to conduct. You asked, Carolee, what inspired me to take on this role as the inaugural director of OADR. And I'm going to share a little bit of my background, if that's okay. So I am a rheumatologist and internist by training. I trained both in the United Kingdom and the United States. So I completed my undergraduate degree at Oxford and then medical school at Imperial College in London. And when I moved to the US, I redid residency and fellowship at Georgetown. I was a physician scientist on faculty there. I studied a number of autoimmune diseases, including scleroderma, lupus, a disease called hidradenitis. And I had a large study investigating the interplay of the immune system and the wound microbiome in patients with chronic wounds. And that included both diabetics and patients with lupus, rheumatoid arthritis, hidradenitis. And then in 2014, I moved to George Washington University, as you mentioned. I was chief of rheumatology there, so I had this kind of tripartite mission, as many academic divisions of rheumatology do. I provided bedside clinical care to patients both in the hospital and in the clinic. I taught our undergraduates and medical students in the field of rheumatology, and I had an active translational research program. But you probably know this metaphor of the person walking past the river and seeing somebody drowning and jumping in to save them and going about their day and then coming back the next day and seeing another person drowning and jumping in to save them. And there comes a point in that metaphor where the right thing to do is to go upstream and figure out why people are falling in. And so that's why I have taken on this role. I'm passionate about autoimmune diseases, passionate about women's health and autoimmune diseases. But autoimmune diseases affect 23.5 million people in the United States, and it's very possible that's an underestimate. To put that into context, HIV at the end of 2021 was estimated to be impacting 1.2 million people in the United States. So this is a huge problem, somewhere between 7 to 8%, lower end estimates in the US, to 10% of the population, based on a recent study out from the UK, of people are impacted by autoimmune diseases. They impact both men and women, but more commonly women. 80% of people with autoimmune diseases are women. And it is really important, as Erica mentioned, to ask why that is, because it's in asking why there are differences that we're going to find answers that drive the science. And that's really why I'm here.
I love how passionate all of you are. It's so amazing to me how engaged you are with your science, and I'm thrilled by that. It's wonderful to hear and wonderful to sense and see. So, Vicki, how does OADR and RWH then plan to incorporate sex-based differences in disease understanding into its research strategies? Because obviously, we all know that 80% number and that 7% even just of rheumatoid arthritis being studied or dollars being allocated. How does this change?
Yeah, I mean, I think that's a really important question. As you know, understanding sex as a biologic variable is very important to the Office of Research on Women's Health. But I think where the Office of Autoimmune Disease is wanting to kind of really make a difference is in helping use the science, use the data to follow the biology and to help us better understand why these diseases happen. Are there places where we could intervene at an earlier stage and change the paradigm in how we approach and manage autoimmune diseases?
Yeah, well, thank you for that. I mean, I'm going to come back to you. I want to ask you all actually the same question, and I'll come back to start with Erica in a minute. So obviously, we are pleased and delighted with the new initiative that the White House has launched to contribute significantly to women's health research. The last number on the State of the Union message was $12 billion. That's a big number. We're hoping that somehow that does get, quote unquote, transferred and become a reality. You know, I'll take a percent of that too. But whatever it is, it's going to have a huge impact. But and I'd be curious to know, Erica, your impression of how that's going to affect you, but also what other entities, such as the business community, the investment community, VC community, academia, who do we need to bring around the table to create this implementation plan of action? Because the money will be one thing, and then figuring out how to allocate that money and what we want that money to do will be another.
Well, the answer is everyone. You know, what's really exciting about this moment right now is that we've now uncovered these differences. Nobody knew they were there before. And if you don't know they're there, you don't have the information you need to understand what the solution should be. It's like we've just discovered gold and we saw a glint of something shiny in a rock. And a couple people started chipping away at it. And the more they chipped away, the more they realized there's a whole vein of gold there. There was so much information to learn. And it directly affects the health of everybody, you know, not just the 52% of the population that's female, but everybody. If you try to make something that's one size fits all, it fits nobody. And the information that we need to help the health of women is going to improve so much for us. If we look at each disease and we compare them by sex, there's two possibilities. Let's say we find that there's no difference. It affects male cells and female cells equally. That's a win because what you've discovered affects 100% of the population. On the other hand, if you intentionally look at this variable and you find that there is a difference, well, that's the information we need. That's the information we need to understand the mechanisms and the causes. As a scientist, all you can hope for is a clear variable and a clear result. If in the cells that have a certain RNA, something happens, and another cell they don't, that's the door you need to follow through. That's the clue you need to find the smoking gun of how disease has developed and where health comes from. And there's a lot we can also learn about how women have a more powerful immune system and how the things that protect women can be supported and strengthened and used to help men. So I think at this point in time right now, it's time for all of us to come together. The researchers, there's information to uncover. There are discoveries that can be made. That's why we get up in the morning. We can throw open the doors of finding treatments for diseases that didn't have them before. For advocacy groups, you know, every disease is different. Let's raise our voices to support research in this area. For businesses, there's an opportunity to fine-tune the treatments that you're making. You know, we don't sell one kind of blue jeans to everybody. We have blue jeans that fit men and we have blue jeans that fit women. We know this. We need immune-based treatments that fit better, that fit the stronger immune response of the female or fit the different immune response of the male. And with all the different groups, whether it's funders or businesses or scientists or clinicians or patient advocates, we can together create that plan of action that we needed. That funding is what creates research. The usual funding cycle is a year or more between when you write that proposal and able to begin that work. We all agree on this panel, we need action now and discovery now. And investment that sparks that research now by families or foundations or businesses allows immediate action and create those opportunities for follow-on federal funding for all the different stakeholders to come together and advance what we need.
Carolee, I would love to follow up on that because, Eric, I love your metaphor of the glint of gold. There's a lot of gold out there that one of the WHAM strategic goals for this year is about mining those data, finding that value that's already there, the study's already paid for, on the private side as well as with the federal government. And I'll add that when we talk about women's health, we talk about diseases that solely or disproportionately or differently affect women. Well, there's a catch-22 with that third point. We don't know what we don't know yet. And so continuing to orient ourselves to these questions about what is the difference by sex and gender can yield so much for the evidence base.
And, people always say bench to bedside, but our approach in La Jolla, and one that's starting to be picked up everywhere, is bedside to bench to bedside, where we invite in the community and the patients that help us understand what the medical needs are and what people are experiencing that has thus far been overlooked by medical science. With that information and that community involvement, we can get to work and use the new tools that science had they didn't have 10 years ago to understand differences between sexes, differences between generations, and differences between diets. And those tools are going to help us uncover the root causes of disease and find the opportunities we need for better treatments.
Well, thank you for that. And thank you, Lori, for the comments, because I think that that is obviously a very important goal of ours. And I know that Erica and the other panelists will be able to help us to figure out how to mine the data, because we're also passionate about this topic that I can't imagine that we won't. But I'd love to ask Montserrat how increased, I'm sure all of us are over the moon, in terms of just the idea that this is being brought to everyone's attention. And how, Montserrat, do you believe that increased funding for women's autoimmune disease research will benefit health outcomes, especially in the research that you're doing, and obviously discovering and exploring more about XS?
Yeah, so I just want to put in a little plug for the importance of basic science research. And the example for that really is the stuff that my lab has been doing. There was no idea in the field of exon activation that immune cells were going to be doing it differently. It was just the dogma that, like, no matter what type of cell you are, you have an inactive X and you're going to maintain the silencing with all the same players. And what we've discovered is that that's not the case, that T cells and B cells do it very, very differently from a fibroblast cell, from an embryonic stem cell. And this is where basic science research, just to understand and to also just the descriptive nature, like what is going on with, in this case, X inactivation in a healthy state, needs to be understood in order to then understand how does it go wrong in the context of an autoimmune disease. And so that's with new fields and stuff, it's so important to get a sense of what the system, how does the system normally work, and then figure out how do these different environmental cell extrinsic factors change the behavior of these different immune cells to start going kind of berserk. So yeah, the funding, I think one of the things that is really striking to me, and I think this came up in this panel, is the fact that there is so much less funding for women's health and for autoimmune diseases. Yet you look at cancer, right? Cancer has a very, very strong male bias. These are cancers outside of the reproductive system, and there's, I think, 5 times the amount of funding for cancer, but there's not as many cancer patients as there are autoimmune patients for that same amount of funding. So I think there absolutely has to be a change in the balance of research funds that address the diseases that we're dealing with.
Well, obviously, WHAM couldn't agree more, and that is a lot of the basis of the data that we accumulated. So, Vicki, how does OADR, which I'll refer to it now, and ORWH plan to identify the emerging areas of innovation in autoimmune disease research? Because obviously, I believe your grant was, or the amount of money that was allocated to your creating your office was $10 million. And I would assume that with this new mandate, there will be sufficiently more dollars that you'll have for your research. Can you talk to us a little bit about some of the insights and resources that you might be using?
So I can't speak to you to this year's budget. I'm reading the same newspaper as you are, but I can speak about last year. And so in FY23, you're correct. The $10 million that Congress appropriated with the initiation of this office. And I can speak to a program that we initiated that I think you all will be very excited about, which is the Exact Plan Awards. So this was a NOCI R21 award that we issued back in spring of 23. They were funded in 23, and actually the funding from OADR, when we are funding projects, we administer those projects through the ICOs. So this was both designed and administered with significant help from NIAMS and from NIEHS and other ICOs. NIAMS definitely carried the bulk of the work for this. But this program, we funded 6 awards in FY23. They're 2-year R21 planning grants to study the interaction of different exposures and the immune system, so the immunome and the exposome across the lifespan. We have a number of projects that were funded, some looking at diabetes, both in children and in adults, some looking at MS, lupus, rheumatoid arthritis, looking across different populations, some looking specifically at some of the pediatric diseases, and one that's specifically looking at skin diseases. But really trying to get to what both Erica and Montserrat had mentioned of, in order to understand the data and in order to really mine the data, we need to harmonize the data. So we need to be looking at both exposures and how we measure immune responses across the lifespan in a way that allows us to use large data sets, existing data sets, and generate hypotheses and answers to those hypotheses from those existing data sets. We are very keen to really think about how do we approach autoimmune disease research moving forward. I want to bring to highlight to your attention a couple of areas where I think we can make a difference, and we can kind of springboard off of things that have worked really well in other arenas. So one is the story for teplizumab, a new drug recently approved for prevention of type 1 diabetes. So complete paradigm shift, but something that could potentially be kind of a similar model for other autoimmune diseases. So in type 1 diabetes, obviously most commonly presents in children. It's a disease organ specific where the immune system is damaging the insulin producing cells in the pancreas. If you are a relative of somebody with type 1 diabetes, you are increased risk of developing type 1 diabetes. And if you are a relative and you have autoantibodies, and there are at least 4 different known autoantibodies that are testable, that predict risk for type 1 diabetes, then you're at very high risk. And what this drug has been shown to do is, if it's given as an infusion, it's a monoclonal antibody given over a 2-week period, one-off course of treatment to people who are at high risk for type 1 diabetes, by which I mean they're relatives of a patient with type 1 diabetes and they have autoantibodies, it can prevent progression to diabetes, which is amazing, paradigm shifting. We knew 20 years ago in lupus, in rheumatoid arthritis, in many other autoimmune diseases, the signal of autoimmunity is present more than 5 years before clinically defined manifestations of disease. And so there is this opportunity, if we kind of shift a little bit of our focus from the approach to disease once it's manifest to how do we actually identify people at risk? How do we understand these triggers that Dr. Anguera was talking about that trigger the immune cells to alter how they're inactivating the X chromosome, perhaps? Some would posit that those may well be exposures such as viral exposures and other exposures that trigger the immune system differently. And could we actually, by studying that phase of pre-disease, understand some of this process of autoimmunity better? And could it lead to new treatments and new interventions for autoimmune disease?
Fantastic. Thank you for that. I think I'd like to turn to a couple of the questions that have been coming in. One, I know a lot of people watching this will be interested in: can menopause hike autoimmune issues? Who would like to take that?
I'll start, and then the other panelists can continue. It really depends on the disease. So every disease is different. Every disease has a different manifestation. Every disease has a different root cause. So some, like SLE, will find their symptoms lessen. Excuse me. Some, like rheumatoid arthritis, will find their symptoms increase. This is the kind of data that we need. If you average all people together, you won't find it. When you separate people by generation and you look at the relative hormone levels, we'll start to find the information that we need to understand if disease is lessening, why. So, one example, in MS, symptoms lessen when people are pregnant. It's remarkable. They feel great. What's going on? Well, the placenta makes estriol, related to estrogen. Maybe that molecule is what's involved in dialing down the switches of the immune system. How do we use that information in order to produce a treatment that's going to lessen symptoms in people who are not pregnant? A lot of information there.
Montserrat, anything you'd like to add to that?
So I just defer to Erica on that. We're not experts in changes in hormone levels. We focus on the X chromosome. So,
right.
But yeah.
Okay. Vicky?
I'm just going to say that I think this is a situation also where a point that was raised by Erica earlier is really important. And I think it's that we need to listen to our patients. And I can tell you, I was a physician scientist. I stood at the bedside of my patients every day. And I think the one truth I can tell you is if you listen to what they're saying and follow the biology, you find exciting science. And I think that's where, we should kind of approach this in a much more inclusive way as scientists, that if there's something to be found and we listen to our patients telling us this is going on, it's usually a good way to find interesting science.
And I would like to amplify that point. What don't we even know to ask, and where can the lived experience be brought in to expand that, in addition to the expert view?
Mature women are sick and tired of being invisible and ignored. And patients with autoimmune disease are sick and tired of going to the doctor and saying, I'm really, really tired. Of course you're tired. You got 4 kids and a job. There's a lot of people that have 4 kids in a job that are not tired. There's something honestly wrong. And these women are sick and tired of having their symptoms overlooked and their disease overlooked and their pain overlooked. We need the diagnostics so we can find the measurement of what's actually going on. And we need treatment so we have something to offer these women.
Totally. And it takes about 9 years, I believe, on average for that disease to be diagnosed, which is not a good thing. All right, it's 3Not30 time. Erica, what do you want to see happen in 3Not30?
I want to see discoveries made available this year so people can do follow-on discoveries next year and the year after that. To make those discoveries, we need to create the funding. We need to create the opportunity. We need to recruit people to the field. We need to raise awareness among researchers to look under our noses at the things that we are doing. If we are using plastic tissue culture dishes, does this create more BPA, more plastics that influence hormones in a sex-specific way? What is the sex of the cell lines that you're using? Do you have enough female samples? Are your female samples premenopausal or postmenopausal? This research needs to happen now. And so we are at, one of the panelists used the term crucible moment of we have the information, let's spark the resources, the funding, and raise the awareness to bring people in now.
Perfect. Montserrat, what would you like to see happen in 3 years, not 30?
From the academic perspective, I would like to see more studies where individuals report the sex of their cell lines, of the sex of their patients, instead of just, glossing it over. I want to see more women in research in academic positions of leadership. I see a lot of women who are starting in graduate school and, fewer and fewer that continue to postdoc, and then even fewer that stay in academia. So how do we change that? So these are things that are on my mind in this academic setting. Like, how do we change what academia looks like, what universities look like?
Great suggestions and great ideas. Vicky?
So mine has to come from science, right? So I think we approach this effort as scientists, and if the immunome is the missing link, then we need data science to integrate these complex data sets to allow us to layer the genetics, the epigenetics, the proteomics, the metabolomics, and the exposures onto the immune system to better understand and analyze human biology.
Excellent. Lori?
Yeah, so what I like about the 3Not30 goals is they're ambitious but feasible. So expanding that evidence base in a way that we've discussed, and then improving how investment decisions are made, bringing the right information to bear on resource allocation once that funding has increased.
Well, you all are so inspirational and so amazing in the work that you're doing, and I'm so grateful for the work that you're doing and that you participated in this dialogue today. And thank you to all of you who joined us to listen to this inspirational conversation. 3Not30 obviously is our call to action, and collectively we hold the economic power to drive significant change. And I think, as Erica pointed out, it's up to us if we want to make it happen. WHAM certainly intends to, with all of your help. With women at the forefront of the economy, making up more than 50% of the population and workforce, we think that this should happen. We have the opportunity to shape a future where everyone thrives, not in 30 years, but in the next 3. So we encourage you to visit WHAM's website at whamnow.org and join us on our 3Not30 initiative to be part of this transformative movement. I think a link went up in the chat with more details on how to follow us and stay engaged in this critical conversation. And of course, we look forward to seeing you at our next dialogue in the series, which is on April 18th, which will focus on women's brain health and how we'll make changes in 3 years, not 30, to impact the lives of women. So let's leverage our collective power to create the changes we envision for a healthier, more equitable future for all. And obviously, listen to the power, the energy, the enthusiasm, the knowledge and the wisdom that this group brought us today. They're there to help support all of the good health that we can all have by investing in research in women's health. So thank you for joining us. We look forward to seeing you in April.